Thank you. And I swear this is the last time you have to hear from me today. Today I'll be talking about I think really timely study in the ALS space. We conducted a large study of serum neurofilament in people with ALS enrolled in the ARC study. And I'll explain kind of why we're focusing on neurofilament right now as we go. But I think it'll be impactful findings. I'm going to start by acknowledging the team members and actually all of the people who've been involved in making this work possible. Alan, who you've already heard from today, Josh, Aaron, Beth, Cindy, Joe, all have been incredibly instrumental in coordinating sample collections and appointments with our ARC study participants, getting the samples in the building, processing the samples, and generating the data, and then organizing the data so that we can make some sense of it. I also want to thank all of our ARC study participants, 215 of whom, in this case, who contributed blood samples, and 215 of whom are living with ALS. Their generosity of just effort and spirit in this study has been incredible, and it really can make a big difference. I want to thank our phlebotomy partners. We have a lot of them who fan out around the country to collect these samples. And all of the sample collection, a lot of it's been supported by Augie's Quest and a CDMRP grant from the Department of Defense. And the quantification of the neurofilament was supported by MT Pharma. They've been great partners in this. So background information. So what are neurofilaments? What is NFL or neurofilaments? So they're cytoskeletal proteins. They're think of them as like structural support for cells, and they make up neurons, not just motor neurons, lots of different types of neurons. And when neurons get damaged, these neurofilaments or NFL will spill into the cerebral spinal fluid and into the blood in a measurable way. We all have neurofilament in us. In fact, like what you're looking at here is a figure from a paper that I pulled that looked at just healthy people over time. You see that babies actually have higher measurable neurofilament in their blood. Then it decreases through adolescence, maybe through kind of like pruning, like you kind of get rid of the neurons that aren't necessarily important during development. They bottom out, and then as we age, neurofilament levels increase over time. Probably just as an aging process, we lose neurons as we age. Why does this matter, particularly in ALS right now? You've heard mentioned today, I think already a few times, a drug called tofersen. Tofersen is an antisense oligonucleotide, so it's a drug that's targeting a specific gene, a specific protein in people with mutations in superoxide dismutase. It is the mutation that my best friend had and caused him to pass away from ALS. This medicine was tested in a clinical trial by Biogen and Ionis. And in April 2023, the FDA approved the drug based on the fact that tofersen lowered levels of neurofilament in people who were receiving drug at time 0 at about 3 months in, and again out to 28 weeks and beyond. And then in participants in the trial who were on the placebo, their levels didn't go down until they went on to the open label extension. So they started to receive the drug at this point, and then neurofilament went down. And that indicated that perhaps in these people receiving the treatment, their motor neurons were dying less. And so the FDA took that evidence along with non-statistically significant evidence of slowing of disease progression as measured by FRS, and they said, okay, we're going to accelerate approval of this drug. And so this brought neurofilament to the forefront in the ALS space as something that could potentially be used in clinical trials for a broader ALS patient population. And in fact, as Paul Wright mentioned earlier during our panel discussion, in Europe, it's already being integrated into at least one platform trial as an endpoint. So we sought to better understand the behavior of neurofilament in NFL. specifically in a broader population of people with ALS. And so the ARC study, where we've been collecting a lot of sample over a lot of time, give us an opportunity to do this. So I'm going to start by describing like the participant subgroup from whom we've collected blood that we measured these samples. So all of these samples were collected, and this kind of relates back to Alan's timeline from earlier this morning. We were collecting these blood samples remotely from people's homes from 2019 through to 2024. And we had lots of different reasons in mind for collecting these samples, and this is just one of the use cases that we're talking about. The phlebotomists do all of the processing on site, so they'll spin the samples and they'll isolate sort of the clear liquid part of the blood called serum and plasma. And then they ship those to us in a cold pack. And then the team at TDI will immediately separate those in the process called aliquot, and you put amounts of the liquid into different vials so we can freeze them for further use where you don't have to freeze and thaw them a bunch of times. And so we handled them that way. As far as the cohort in general, like the people who were in this, like I said, it's 600 samples from about 234 people. In general, we were getting about 3 samples per person, if you look at it that way. 215 of them had ALS diagnoses, and about half men, half were women. We did have 15 asymptomatic mutation carriers. So these are people who had like a SOD1 mutation or a C9orf72 mutation, but no disease symptoms at all yet. And then we had some healthy volunteers as well. The ages of these individuals do tend to represent what we see generally in ALS. We certainly had representation from people who are very, very young, people who are quite old, and also people right around where you would typically expect sort of your average ALS diagnosis in the 50s and 60s. However, it's just worth noting our ethnic and racial distribution does not represent the general population, and that's something that we and really the entire field need to get better at, and we are looking at ways to do that. One point that I want to make about the group that we looked in is we were definitely, this is probably a little bit different from most ALS patient cohorts in one way. We had a lot of people where we were collecting samples long after their symptom onset. Right? So we certainly had people where we were collecting samples within 2 years of onset, which is often what we're looking at in clinical trials. But we also had representation from people where we were collecting 10, even 15 years after onset. So that's interesting to us to see what's happening in neurofilament in people who might be very far progressed and stable, and to see if we can get a sense of that. But that also means that we're collecting a lot of samples from people who tended to be slower progressors, right? So people who were not losing as many FRS points. So what you're seeing in this figure is we have a typical ALS clinical trial is tending to enroll people who are losing like 1 FRS point per month. In this study, we had a lot of people who were losing like a half an FRS point per month. We also had people who were faster progressors who were losing more than 1.5 points, 2, 3, et cetera. But we did have, we were enriched for slower progressors. That's partially because of the way our ARC study works. We enroll everybody. Everybody with ALS can join the study. People who are slower progressors tend to live longer, and they have more time to find us and join the study, and we welcome them into the study. So what did we do? We were measuring neurofilament, and we did it through 2 different research vendor services. So there are 2 prominent vendors of this right now, at least in the ALS world. One is a company called LabCorp, and one is a company called Quanterix. And I've heard data reported back from both of these platforms. We wanted to know how they behaved when we compared them to one another. They use 2 great technologies that are quite different. The LabCorp technology kind of uses an electrical current, which ends up running through a label that emits photons, and they were able to count the specific number of molecules by the number of photons emitted. And the Quanterix assay, they actually use imagine a little tray with tiny microscopic holes, and each hole is big enough to hold 1 bead. 1 bead will bind to 1 molecule of neurofilament. So the number of beads filling a hole that light up in that assay tell you how much neurofilament is in the sample. So two very different approaches to kind of measure the same thing, but both very high quality, such that both were designated by the FDA as breakthrough devices for the use in multiple sclerosis in the past year. So we needed to use we needed to kind of do it two different ways depending on the company. They had different preferences. So LabCorp asked us to send 12 batches of 50 vials over a month and a half. They asked us to let the samples go to liquid. They thawed out, and then we shipped them at room temperature. Quanterix wanted all of them at once, 600 samples at once on dry ice so they could run them all at the same time. And this was all massive effort, actually, logistically, by Josh Berman and Alan Primaseri to get these samples out. So what happened? The first thing we did freaked me out. We looked at how the levels compared across the two assays. You kind of would have expected them to say the same thing, because you're measuring, in theory, the same thing. But the results were quite different. The average level of all of the samples from the Quanterix platform was about 44. The average level of neurofilament as defined by LabCorp was about 8. So we were kind of worried, like, is one of these wrong? Are both of these wrong? How do we know? Then we made the next obvious assessment, like how do they compare directly to each other? So each dot here represents a single sample. And so what we learned was that if the levels were high in the Quanterix platform, they were also high in the LabCorp platform. They were just on different scales, right? And do I know what the absolute value is, what the right one is? No. I don't know. I don't think anybody does. But it might not matter, because if you're measuring within the same platform and you're making comparisons, that's all that really matters, right? And when we did, so if you look here, the relationship is about 5.6 times. So Quanterix levels were about 5.6 times the LabCorp levels. So 200 relates to about 37. And 100 relates to about 17 or so. And when we looked more closely at the sort of the package insert of the Roche assay from LabCorp, they had done this in non-ALS samples, and they actually saw the same thing. So when they were looking at samples from people with no disease or other diseases, they saw the same exact relationship, that 100 relates to 17. And 200 relates to about 37. So in retrospect, it wasn't surprising. But in my conversations with people in the ALS clinical community, this isn't well known. So if you're getting your neurofilament levels reported back to you, you really need to know which assay system that's reporting. Because if you get read back 100 and it's coming off of the LabCorp results, that's really, really, really high. Whereas if you're getting, a level reported back of 30 and it's from the Quanterix, well, that's relatively low. So it is important to know, and it's important for your clinician to know which assay system they're looking at. So what did we learn when we were actually trying to dive into what does this tell us about ALS? So our preliminary findings. Well, the non-surprising thing is, yes, people with ALS had much higher levels than people who were healthy. Or asymptomatic mutation carriers. Asymptomatic mutation carriers and healthies looked pretty much exactly the same. And this is all in the Quanterix data now that I'm referring to. All slides going forward will be in the Quanterix platform. So not surprising, but very good to know that we validated what's generally expected. A next question we asked was, are levels different as a function of how severe your symptoms are at any given moment? So we're looking at FRS on the horizontal x-axis, and we're looking at neurofilament levels on the y-axis. And so what you're seeing here is that people who have less severe symptoms, so higher scores on the FRS, had tended to have lower levels of neurofilament. But that was not a very strong relationship. And it might just be because people who have higher scores are more likely to be slower progressors. So they have a longer time to have higher scores. We looked for if there were any relationships, any differences between men and women. None. Basically the same. As a tiny trend for women to have slightly higher levels. I mentioned earlier that people, just healthy people, tend to have higher levels of neurofilament as they get older. That effect is essentially washed out in the ALS population. So at least in our cohort, it really didn't, maybe a slightly higher trend, and maybe if you had many, many thousands of samples, you would be able to discern a relationship between age and neurofilament in a cohort of people with ALS, but it really isn't the primary driver here. What did hold is what one would hypothesize, right? That people with faster progressions would have more neurons dying, and so more neurofilament would spill and be detectable in the blood. And so what you're seeing here is these people are the faster progressors. They're losing more FRS points per month, and so they have higher levels. And in each of these categories, we do see a statistically significant difference. And again, we have a lot of blood samples that were collected from people who were slow progressors, and so you have more dots there. But it was still statistically significant at all of levels. But I want to point out that, yes, you can be a very slow progressor and have levels in line with people who are very fast progressors. And you can be a very fast progressor and have levels that are in line with people who are slower progressors or asymptomatic. And that's a little bit surprising, and we really want to understand these outlier cases so we can make sense of it if we're using this in clinical studies in the future. So one other question that we're still in the process of asking a lot of questions here, but this is another one that we dove into. We asked about symptom onset. So we talked about how different people have different regions where they're affected more early on in disease. So people who are bulbar onset, so neck, throat, swallowing, etc., had higher levels than people when we were measuring neurofilament than people who had limb onset or had onset in both. That might be because it's general consensus that people with bulbar onset are faster progressors. So that might be what's driving it. What might also be driving it is that when we're drawing the blood samples from them, they've actually progressed to a point where they're affected in their limbs at that point. And perhaps that's actually what we're capturing, that larger motor units are dying and we're able to measure those levels. We need to better understand that, but it's interesting, and we can dive into that because we do know about their symptomatic status in their different regions at that point in time. A next question that we asked is, are neurofilament levels stable in people with ALS? There's a model that has been proposed, and I think there's some reason to support it, that levels are low, right, before you have symptoms, and at some point as you start to have disease onset, your levels climb, and at some point they reach a plateau, and maybe that plateau comes when you're clinically manifest. And the reason this matters is because if the levels are stable, then you would have an easier time measuring a drug effect, right? So if a drug is protecting your neurons in the context of stable levels, if your amount of neurofilament goes down, it might indicate that fewer neurons are being damaged and you're measuring that appropriately. So we asked that question a few different ways. The first way I asked the question was, I looked at people from whom we collected multiple samples, and I asked, what's their change in FRS score from their first draw to their last? In people who had a very big change in their FRS score, so these are people who lost like 28 points, or this person who lost 26 points during the window where we were drawing samples, was there also variation in their neurofilament that was measurable during those blood draws? And yes, there actually is more variation in people who have more change in their disease state, which suggests it may not be completely stable. Like if you have a more dynamic change in your disease state, neurofilament might change as well. But generally speaking, in this figure, we're actually looking at percentage change from your baseline blood draw as a function of time since your disease onset. And these are little blocks of months here. Generally speaking, the average tends to kind of resonate around 0, that there's very little change. But if you look at each individual person's blood draw, there's a lot of variability in that. It can be very bouncy. And I think it's really important for us to understand that bounciness when we're designing trials so that we can have a better sense of how to statistically power it, or how to select time frames that we're trying to measure this to look for drug effects. So I'll just summarize. So in general background, like serum neurofilament is a good generic indicator of neurodegenerative processes, and nothing that we saw would dissuade me of that. Changes in serum neurofilament was useful for advancing tofersen into accelerated approval. The ARC study provided a really great opportunity to make sense or make better sense of this potential biomarker. It's an incredible asset, and I think we can use it over and over again for other outcomes as well, other measurements in blood. The two leading assays platforms that are used to measure neurofilament report back very different values. And it's not very surprising if you look at their history. But they share essentially the same information though, because the relative values within each one holds up. And so it's just really, really important to know the one that you're looking at when you're making interpretations. LabCorp, really cool, in my opinion. There were no batch effects. Every single batch, all 12 batches kind of lined up on that same correlation graph the way you would hope. I can't make that same determination for the Quanterix set because we did it in one batch, so I have no idea if a second batch would be different. So serum neurofilament levels higher in people with ALS. Serum neurofilament levels are higher in people with faster progression. Serum neurofilament levels in our cohort were higher in people with bulbar onset. And while the levels tend to be stable, they can vary more in people who have more dynamism in their disease progression. So I think that serum neurofilament is really going to be a very useful prognostic biomarker for people with ALS in the clinic, and also as a measurement of clinical therapeutic effect. But I do think that this indicates that more needs to be done to really hone in on what might drive those big spikes or those big dips in neurofilament that's measurable. And so what's next for us? Well, this is it, right? So sharing the data with the ALS research community and the clinical community, and really having conversations with neurologists about like, well, what are you seeing? And how does what we see here relate to what you see clinically? And a careful evaluation kind of in partnership with those neurologists about outlier cases, right? So why do those spikes happen or those big dips happen? And then we'd like to craft a follow-up study after we've had a chance to parse the data and really craft different hypotheses to test it, right? We can do that through the ARC study. We can look at our cohort. We've been collecting way, way more samples all the time. I think we're now up. When we started this study, we had the 600 samples. We took every one out of the freezer and tried it. Now we're up to, I think, like 900, and that's just a few months later. We'd like to work toward established guidance for best practices on using neurofilament for clinical prognosis and clinical trial endpoints. And then you've heard from Alan that we have accelerometers and voice recording data, these digital outcomes. We kind of want to relate these neurofilament data to those digital outcomes and see if maybe there's actually a richer relationship there than we see with ALS functional rating scale. And I won't go into these. We have actually started doing some of that work, but I won't go into that. I know I'm out of time, so I'll go to questions.
Hi, Fernando. You talked about the NFL being a good prognosis, I guess, biomarker. Is there hope that that can be a diagnostic biomarker as well? And also, do you see there being, is there a chance NFL won't be as highly sought after because of those outliers you're seeing, where maybe it won't be accepted as an industry standard because of outliers, or is that not a concern?
Both great questions. I think it's hard to imagine it as a diagnostic biomarker all by itself because like you see elevated neurofilament as a function of lots of neurodegenerative conditions. You see it as a function of a concussion, right? Like you can measure higher levels. In fact, some of these outlier cases might be a person who had a fall. And so we have to, like, that's why you have to look really carefully at those other data around those samples that look different, like what might explain it, because that might teach you how to use it. I think it's going to be useful, and it'll probably be in the constellation of things that are used in clinical trials over time. Studies like this one, like looking at the natural history, will teach us how to do it. We're not there yet, we're not done, but I think this is a step in the right direction for that.
So I'm Meg Reyes, and I actually had a couple questions from the previous one, so I'm going to kind of ask them because it kind of ties in.
Okay.
So when you do your study, do you define it by non-gene holders and random, or okay? So do you find that different in your studies when you're looking at the NFLs?
Great question. We do define it. I haven't looked at that yet. So the data are like 2.5 weeks old.
Oh, okay.
So, no, it's great. Like that's definitely high on the list of questions. And so you should be a scientist.
So I wish. I was just posting that. I needed to study better in biology, and because this is, whoa. Anyhow, the other thing is that I know we talk about ALS being a rare disease, but we also talked about how many people have ALS, that, or even it's kind of like hypothetical sometimes because we really can't get there. And recently I've had a lot of people ask me in like caregiver groups that their spouse was diagnosed with PLS, not ALS. Mm-hmm. So in your studies, and you just said that your NFLs, you could see it with concussions. Do you see any of the PLS? Do you even look at any of that as a correlation at all?
So we definitely keep track of people who tell us that their initial diagnosis was PLS, and a lot of times that can kind of convert to ALS. So we definitely look at that. Right now, that's really the main way that we're able to look at sort of the exclusive upper motor neuron symptoms. We do have a survey that tries to capture some of that too, but it's actually something we can do a better job of, and I'm glad you brought it up. Okay, thank you.